Welcome to Lesson 20.2, Prokaryotes. In this lesson, we'll explain how prokaryotes are classified, we'll describe how prokaryotes vary in structure and function, and we'll explain the role of prokaryotes in the living world. Microscopic life covers nearly every square centimeter of Earth. The smallest and most abundant of these microorganisms are prokaryotes, unicellular organisms that lack a nucleus. Prokaryotes have DNA like other cells, but their DNA is not found in a membrane-bound nuclear envelope as it is in eukaryotes. Prokaryote DNA is located in the cytoplasm. For many years, most prokaryotes were simply called bacteria. We now know that the classification of prokaryotes is more complex. Prokaryotes are classified as bacteria or archaea, two of the three domains of life. These groups are as different from each other as they are from eukaryotes. The domain bacteria corresponds to the kingdom Eubacteria. The domain archaea corresponds to the kingdom Archaeobacteria. Bacteria is the larger of the two domains of prokaryotes. Bacteria include a wide range of organisms with lifestyles so different that biologists do not agree about exactly how many phyla are needed to classify this group. They live almost anywhere, in fresh water, in salt water, on land, and on and within the bodies of humans and other eukaryotes. This is an illustration of E. coli, a typical bacterium that lives in human intestines. E. coli has the basic structure typical of most prokaryotes. Bacteria are usually surrounded by a cell wall that protects the cell from injury and determines its shape. The cell walls of bacteria contain peptidoglycan, a polymer of sugars and amino acids that surround the cell membrane. Some bacteria, such as E. coli, have a second membrane outside the peptidoglycan wall that makes the cell especially resistant to damage. Some prokaryotes have flagella that they use for movement, or pili, which in E. coli serve mainly to anchor the bacterium to the surface or to other bacteria. Under a microscope, archaea look very similar to bacteria. Both are equally small, lack nuclei, and have cell walls. There are important differences between bacteria and archaea. The walls of archaea lack peptidoglycan. The membranes contain different lipids. The DNA sequences of key archaea genes are more like those of eukaryotes than those of bacteria. Based on these and other observations, scientists have concluded that archaea and eukaryotes are related more closely to each other than to bacteria. So how would a diagram of an archaean be similar to the diagram of the bacteria shown on the previous slide? Well, like the bacterium, the archaean would lack a nucleus. It would also have a cell wall. How would a diagram of an archaean be different from the diagram of the bacteria shown on the previous slide? The cell wall would be labeled differently because archaea do not have peptidoglycan in their cell walls. Archaea live in extremely harsh environments. One group of archaea produce methane gas and live in environments with little or no oxygen, such as thick mud and the digestive tract of animals. Other archaea live in extremely salt environments, such as Utah's Great Salt Lake, or in hot springs where temperatures approach the boiling point of water. Prokaryotes come in a variety of shapes, though there are three basic shapes. Rod-shaped are called bacilli. Spherical prokaryotes are called cocci. Spiral and cork-shaped prokaryotes are called spirilla. You can also distinguish prokaryotes on whether they move and how they move. Some prokaryotes do not move at all. Others are propelled by flagella. Some glide slowly along a layer of slime-like material they secrete. Prokaryotes obtain energy in diverse ways. Prokaryotes need a supply of chemical energy, which they store in the form of fuel molecules such as sugars. Take a look at this table, which compares the way prokaryotes obtain energy. Some species are able to change their method of energy capture or release depending on the conditions of their environment. What is the term for a prokaryote that uses only light as its energy source? A photoautotroph uses only light as an energy source. Prokaryotes release energy in diverse ways. Energy is released from prokaryotes' fuel molecules during cellular respiration, fermentation, or both. Take a look at the table, which compares the ways prokaryotes release energy. Which modes of metabolism release energy through fermentation? Obligate anaerobes and facultative anaerobes. When a prokaryote has grown so that it has nearly doubled in size, it replicates its DNA and divides in half, producing two identical cells. Because this process, binary fission, does not involve the exchange or recombination of genetic information, it is a form of asexual reproduction. When conditions are favorable, prokaryotes can grow and divide at astonishing rates. Some divide as often as once every 20 minutes. Prokaryotes reproduce asexually, so how do their populations evolve? They do so by mutation and conjugation. Mutations are one of the main ways prokaryotes evolve. Mutations are inherited by daughter cells produced by binary fission. Many prokaryotes exchange genetic information by conjugation. During conjugation, a hollow bridge forms between two bacterial cells, and genetic material, usually in the form of a plasmid, moves from one cell to the other. Many plasmids carry genes that enable bacteria to survive in new environments or to resist antibiotics that might otherwise prove fatal. This transfer of genetic information increases genetic diversity in populations of prokaryotes. What is the result of the processes of mutation and conjugation? These result in genetic changes in prokaryotes. Are mutation and conjugation forms of reproduction? No, these processes are not forms of reproduction. They do not result in the formation of daughter cells. When growth conditions become unfavorable, many prokaryotic cells form an endosperm, a thick internal wall that encloses the DNA and a portion of the cytoplasm. Endospores can remain dormant for months or even years. The ability to form endospores make it possible for some prokaryotes to survive in very harsh conditions. What type of organism is vital to recycling matter in an ecosystem? That's a decomposer. Bacterial decomposers are also essential to industrial sewage treatment, helping to produce purified water and chemicals that can be used as fertilizers. Bacteria of the genus Rhizobium often live symbiotically with nodules attached to the roots of legumes such as clover, where they convert atmospheric nitrogen into the form that is used by plants. Cyanobacteria, some types, form filamentous chains in ponds and other aquatic environments where they perform photosynthesis. Bacteria called actinomycetes are present in soil and in rotting plant material such as fallen logs, where they decompose complex organic molecules into simpler molecules. The growth of organisms such as bacteria can also disrupt the health of ecosystems. For example, an oversupply of nutrients causes the explosive growth of algae and other producers in freshwater lakes. As these organisms die off, bacterial decomposers flourish. When this happens to excess, the bacteria can use up virtually all of the dissolved oxygen in the water, causing massive fish kills that further disrupt ecosystems. What type of organism is found at the base of every food web? A producer. Photosynthetic prokaryotes are among the most important producers on the planet. One example is a tiny cyanobacterium, which alone may account for more than half of the primary production in the open ocean. food chains everywhere are dependent upon prokaryotes as producers of food and biomass. While nitrogen gas makes up 80% of the Earth's atmosphere, only a few organisms, all of them prokaryotes, can convert nitrogen into useful forms. The process of nitrogen fixation converts nitrogen gas into ammonia. Ammonia can then be converted to nitrates that plants need, helping to maintain healthy ecosystems. Nitrogen-fixing bacteria and archaea provide 90% of the nitrogen used by other organisms. The rest is provided by nitrogen-containing compounds from weathering rocks. Some even comes when lightning combines oxygen and nitrogen in the atmosphere. So how do humans and other animals get the nitrogen they need to make proteins? They eat plants or animals that have eaten plants. Plants get nitrogen from nitrogen-fixing bacteria. Some plants have symbiotic relationships with nitrogen-fixing prokaryotes. The bacterium Rhizobium grows in nodules or knobs on the roots of legume plants such as clover and soybean. The Rhizobium bacteria within these nodules convert nitrogen in the air into the nitrogen compounds essential for plant growth. In effect, these plants have fertilizer factories in their roots. Let's review the key points of this lesson. Prokaryotes are classified as bacteria or archaea, two of the three domains of life. Prokaryotes vary in size and shape in the way they move and in the way they obtain and release energy. Prokaryotes are essential in maintaining every aspect of the ecological balance of the living world. In addition, some species have specific uses in human industry.